to start with just saying I don't really actually do fMRI research, but I read a lot of it, so I think it's really interesting stuff, and I'm excited to be able to talk to you a little bit about the journey that has taken us from sort of early electrophysiology studies in the 90s on sleep and memory that have moved us into the human brain and looking at how we're actually encoding memories during consolidation periods of sleep using fMRI.
So I want to start with this quote by Riverside. If sleep is an absolutely vital function, it is the greatest mistake evolution's ever made. And that's Riverside, who basically coded all sleep stages and that we use every day in my lab to say what the different stages of sleep are. And what he means by this, obviously, that we spend a third of our time sleeping and very little of it, very little of our waking time researching it. We don't really understand what's going on, but clearly there must be something important for why it hasn't been sort of evolutionary fixing the curve where we still sleep so much. Another funny way of talking about sleep is it's the biggest embarrassment of neuroscience. But I think that is not necessarily the case anymore, because now, as you can hear from Jesse, that there's some really interesting, exciting work going on in neuroscience looking at sleep. But for a long, long time, sleep was a completely dormant field in terms of neuroscience. So I'm just going to begin by telling you a little bit about, why we might want to study sleep, and then talk a little bit about what sleep is and how we look at it, and then
take the journey into the relationship between sleep and memory. Okay. So the National Sleep Foundation reports that we're sleeping less than we need to. We should be sleeping around 8 hours, and people mostly are getting less than 7 hours of sleep. And this is true for both men and women. Women recently reported that 67% of women reported trouble sleep more than 3 nights a week, which is really a shocking amount of sleep deprivation for women. And of course, a lot of things happen in a woman's life, and in particular, going through having children, of course, and this happens to men as well, but that can be a huge impact on sleep, and also for as women age, they also have changes in their sleep patterns. This is also true for, that you see this across different age groups, and that sleep has been shown to be important across different age groups, so much so that even a study showing that infants who are not sleeping as much as their peers have a greater chance of becoming obese as adolescents. And this is also true when you look at adolescents. There are also behavioral problems that go along with decreased sleep, such that if you actually look at all the kids who are labeled as ADHD, a lot of them actually are just not sleeping enough. And when you get to, there's a study that looked at just getting people to sleep more, getting adolescents to sleep more, and then their rates of ADHD diagnoses actually decreased. So behaviorally, there's also impacts of sleep. And of course, we know that as we get into older adulthood, sleep changes a lot, and our cognition changes a lot. And there's very, very little research right now, it's a very new field, looking at whether some of those changes in sleep correspond also to those changes in cognition. Okay, so what's the cost of not sleeping enough? Well, there's lots of, I've already mentioned some of those costs, but there's cost to our health where sleep restriction or sleep deprivation leads to an increased risk for heart disease, obesity, diabetes, stress, and depression. There's all sorts of safety concerns, right? And this is pictures from the Exxon Valdez, where one of the person actually at the helm at the time of the accident hadn't slept for like 48 hours, and the main captain had actually passed out from drinking. So it was a bunch of things that went wrong on that day. But one of them was that the guy wasn't sleeping enough. And of course, you've also heard about the air traffic controllers, who've been falling asleep in all these horrible accidents from that. The Department of Labor says that actually the fact that we're entering into work every day with a level of sleep, with all the sleep disorders and the lack of sleep, that we're entering to work every day with a 4% loss in productivity. And of course, what we care about at the university's situation is how sleep is affecting our cognition. And there's all sorts of studies looking at adolescents and college students showing that if you are not getting enough sleep, you're falling asleep in class, you're not doing well. And also, of course, what we care about in the laboratory is how sleep can affect our memory and attention and other skills, decision making. So that's obviously just giving you the broad strokes about why is we sleep. And so now, what is sleep? So I'm going to give you just a really brief overview. I just want you to focus on this little section down here. So there's different stages of sleep, and we go through all of our stages of sleep within a 90-minute cycle. So we have these 90-minute cycles all through the night. I'm going to sort of show you what the EEG pattern looks like after this one. So first, we go through a period of early stage 1 sleep, which is just kind of really falling asleep. That period where you have those kind of myoclonic jerks, and it lasts for about 2 to 5 minutes. And then you have a long period, about half an hour of stage 2 sleep. And stage 2 sleep is just the brain just beginning to really get into a sleep state where you show slowing of the electrical signals and specific features of sleep called sleep spindles and K complexes. I'll show you what those look like. The next half an hour of this 90-minute cycle is usually mostly slow wave sleep, which is when the brain is actually synchronizing to a very slow delta rhythm. It's anywhere between 0 to 1 to 4 hertz, basically. And the cortex as a whole is basically showing these very large signals of delta rhythms. Then you pass through stage 2 again, and you go into what's called rapid eye movement sleep for the last half hour. So these are kind of broken down in a half-hour segment, half-hour segment here, and half-hour segment here. Rapid eye movement sleep is sort of the famous sleep stage that is when we have our dreams, and it's also when we have these rapid eye movements. And interestingly, it looks very similar to waking. Actually, the EEG looks very similar to waking because your brain is actually very, very active. But but your body is very inactive. You have paralysis, so you don't act out your dreams, which is a positive thing. And so this is sort of the platonic ideal of the sleep cycle, is in fact broken up into these half-an-hour segments. But if you look up here, the night is actually a little bit different. These are color-coded so that the blue is slow wave sleep and the red is yellow. And as you can see, in the first part of the night, you have a lot of slow wave sleep. And then as you go through the night, then you begin to have less and less slow wave sleep and more REM sleep. And that's because across the day, you build up your need for sleep. It's called a buildup of sleep pressure. And what that is actually your need for slow wave sleep. And so if you go to sleep early in the part of the night, the first 90-minute cycle, these bars denote the different sleep cycles. The first sleep cycle is going to have a lot of slow wave sleep and not a lot of REM sleep. And then as you satiate your need for slow wave sleep, the last cycle of the night is going to have mostly REM sleep, and that's when you have a lot of your dreams. So if you woke up, you feel like you were dreaming all night. It was really actually that last cycle that seems to be important. And a lot of interesting studies have shown that, so as you can imagine, if you're looking at sleep and memory research, and you want to look at the effects of specific sleep stages, one way you could do that is by testing people on comparing their early night sleep to their early morning sleep and seeing how that changes their performance on specific tasks. Another way to do it is through napping studies, but for the first time in my life, I'm not going to mention napping in an entire study. I think I just did, but that'll be the last time I mention it. Okay, so how does EEG look? So here we have the EEG from waking, as you guys can see over here. What you find is that when your eyes are closed, you see a lot of alpha rhythms, but when your eyes are open and you're awake and looking around, you see a lot of what's called beta rhythm, sort of very high frequency, low amplitude activity. And then as the brain starts to fall asleep, you head into stage 1 sleep, then stage 2 sleep, stage 3, stage 4, this is when you're into deep slow wave sleep. So things that you can notice here is that in stage 1 sleep, you have what's called theta rhythms. So just the general thing that you can notice, because it's kind of impossible to sort of learn what EEG looks like in 1 second, but there's a general slowing down, right? And as you go from stage 1 sleep to stage 2 to stage 3, you really start to see that the cortex is really synchronizing to this very slow rhythm across the entire brain. Certain aspects of stage 2 sleep are really important. I'm going to be pointing out a little later is sleep spindles. It basically looks like it's a very slow, it's a very fast little, little sleep feature, which basically goes between 12 and 16 hertz, and it looks like a little spindle. It's a waxy wing. Yeah, this is not a perfect spindle, but anyway. And these are K complexes, which are also features of stage 2 sleep that help us kind of stay asleep and keep us deeper and deeper and deeper into sleep. Okay, and so, and this is just sort of showing you exactly what I showed before. This is what's called a hypnogram, where you look at, where you can see all the stages of sleep. This is the nights of sleep, so this is the nighttime and morning. And as you can
see there's more deeper stages 3 and 4 sleep, slow wave sleep in the first part of the night, and then a lot more REM sleep in the last part of the night.
Okay, so I was asked to talk about topics in sleep. There's a lot to talk about, obviously. It's an entirely large range of topics. So I think, obviously, we can talk about basic neuroscience and just say, well, what goes on in the brain during different stages of sleep. That could be quite interesting. Another topic is to talk about sleep and cognitive function, specifically about memory consolidation, which is something that I find very interesting. But and then another topic would be to talk about clinical questions, such as what happens in sleep disorders. Insomnia is a really interesting thing to study because a lot of the time when you look at insomnia patients, they're actually not sleeping that differently with the EEG output. But what you can see is using fMRI, could you look inside the brain and see actually are there different compensatory mechanisms going on during in people with insomnia, people with insomnia versus people without insomnia. Same thing with aging, changes in sleep occur with aging. How is it possible that people successfully age while their sleep is decreasing or cognition is decreasing? What are the neural mechanisms underlying that kind of successful aging? And of course, sleep deprivation, some of the studies going on here at UCSD from Sean Drummond's lab, looking at sleep deprivations and other compensatory mechanisms of sleep deprivation. So there's lots of different topics to talk about sleep, but I'm just going to focus on sleep and cognitive function. Because I think that actually that is the area that fMRI has a large potential to really make a really exciting impact right now. But what that would require would really be to first get a lot more advancement in terms of the methodological uses of fMRI for sleep research. And some of the stuff that we talked about today just sounds so exciting, and they're not really being used for sleep research. So I think there's a large open field of questions that could be asked. And also, for me, the really interesting thing would be to start answering the question of how do we link the electrophysiology research from animals on sleep and memory to the bold signals that we show that look very, very similar, but are obviously tapping into very, very different mechanisms. And so what is the overlap there? Okay, so I'm going to talk about, I'm going to begin with a study that was done by Wilson and McNaughton in 1994. And this was the study, I'd say, that launched 1,000 ships. It was really the study that came before any of the big flashy research on sleep and memory in human behavior. And it was showing that if you have animals, basically they were doing electrophysiology recordings of CA1 place cells, and place cells are neurons that code for spatial position. And you can actually track an animal tracing through a maze by just tracing their place cells. And what they were recording from 40 different place cells. They had the animals sleeping before they did the maze run, and then they did a maze run for 20 to 40 minutes, and then they did a post-sleep right after the maze run. And they were recording from the same neurons. And this is the neurons in a circular array like this. This is all the neurons that were recorded prior to the run. These are the place cells that were recorded during the run, and these sort of bold red lines indicate high correlations between pairs of place cells. And then the animals went to sleep. And what you find is a very strong reactivation of the pattern that they found during the pattern of place cell pairings that they found during the run. And the idea is that during this period of slow wave sleep, actually, that the animal was somehow reactivating these memories or reactivating that experience. And this was a really important idea because it was the first evidence that we actually are doing that something from our daytime experience was coming into our nighttime experience, and that maybe that was some key to understanding how we actually consolidated memories. And these studies and further studies from the same, from Matt Wilson's lab, is a beautiful other study where he looked at coordination of this kind of replay activity in the brain during sleep between both the hippocampus and also the visual cortex. So here's the place cells, they were sort of running in this figure 8, and here's the CA1 cells and visual cortex cells. And here is the during this post-sleep. And what you find is a really beautiful, much sparser coded and actually faster, but similar representation of the same sequence of cells coding these place sequence cells, coding the run as also going on during post-run sleep. And you find the same thing going on with the hippocampus here during the run and also the post-run sleep. So the question then became, well, how do we do this in humans, right? So and this is also at the time, right around, I guess like that was, the first paper was 1994, and really the big sleep and memory human papers started coming out. There was one in 94 as well, which was by Carney and Sagi, and they were showing that REM sleep seemed to be important for visual perceptual learning. And then Bob Stickles started coming out with all these really beautiful studies on that same task and really trying to look at, well, what about sleep? So looking at the first part of the night with a lot of slow wave sleep, second part of the night with a lot of REM sleep. What's the relationship between that sleep and the performance changes that they saw? But it really took 10 years before we developed a task and developed a methodology to actually start looking at human sleep and memory. And the first one was actually done in PET. And so, and you can see they were really borrowing from the electrophysiology studies about maze exploration, in terms of humans. So they created this maze, which is like a virtual maze, and they had to sort of explore this little city. And they were doing a trial, they were doing a maze. And the question was that if they let the subject fall asleep using PET, what could they find in terms of reactivation? And here, this is performance prior to and after the maze learning. So the gray is dark and white. Gray is the pre and white is the post, just showing that there's actually improvement in time. And this is actually overnight improvement by hippocampal activation and the encoding. So it's actually not really replay, but it's just sort of saying that the encoding of the hippocampus was correlated with overnight improvement. But the really exciting part here is that the same hippocampal brain areas that were activated during encoding were also, there was increased blood flow during consolidation period as well. So this was the first study that kind of was beginning to show the same kinds of results of, is this what we would call replay? And the question is, can we get finer grain analysis of this when we go into fMRI? And so the second study that I want to point out, there's a series of studies that we're going to go over, and just to talk about, what is it that people have done now to improve on this methodology, and what were the first kind of studies that were showing this. So the next study I want to talk about is by Yatsumoto, and they were using this. It's really hard to see, but this is a texture discrimination task, which is a visual task where you have to discriminate the orientation of targets in the periphery while you're staring at a central fixation. And this is the task that had been previously shown by Carney and Sagi in 1994 to depend on REM sleep. And there's a lot of studies that I've been using, and I actually use this in the scanner as well for one of my studies. So it shows really beautiful results because it's retotopically specific learning, and it's great for the scanner because that means that if you have a target, you're learning a target in one retotopic field. If you switch the target to another retotopic field, you're not going to show a transfer of learning, and then you can show what is the neural activation in the trained versus the untrained visual field. And so they were adapting that idea in order to look and see, well, can we actually see the same trained, untrained neural activity during sleep post-learning? So the way we did that, so the first question is, can people sleep in the scanner? Obviously, is a big question. It hasn't quite, I guess Jesse was talking about a study where they had all sorts of long periods of sleeping in the scanner and then waking people up. And it's actually more difficult than you would think. I think people fall asleep in the scanner all the time, but not intentionally. And the hardest thing is that you have to keep still, while you're in the scanner. So how do you manage that? So there's definitely some methodological issues there that need a lot of looking at. So the way that they dealt with that is that they had 2 nights of adaptation sleep. So they had people sleeping first in a mock scanner and then a real scanner just to get them associated with sleeping in the scanner. Then they had, and these people are really only sleeping for 40 minutes. So that first period of sleep, which is mostly non-REM sleep, they really only are caring about non-REM sleep because all the animal research is really also just focused on non-REM sleep. So none of the research I'm talking about is REM sleep dependent. So they would let people fall asleep in the scanner here, and they would record what was going on in their visual areas, and they did an ROI analysis of both the trained visual field of primary visual cortex and the untrained visual field of the other side of primary visual cortex. And then they were, in the daytime, the next day, they were trained on the textual discrimination task on one, with a target in one particular region. And then they had their post-training sleep the same period of time they were asleep. And then they compared that to people without training. So what did they find? Well, first they did a subtraction between pre-training, this is trained, untrained areas of visual cortex, so they're just looking at primary visual cortex. Pre-trained areas, at pre-training there was no difference between the trained and the untrained areas of visual cortex, but post-training they found that in area B1 there was a significantly higher activation during sleep of that same area that had already been processing the target. And this is a very poor, this is a very supplementary, so it doesn't look very good, but this is just showing that this is, that red atopically labeled early visual areas, and they're showing this activation during slow wave sleep of the same visual area that was processing the target during wake. Okay, so this was kind of a landmark study. Showing that we could actually really identify activity during sleep at all. And another study that came out, which was also a really exciting landmark study, was by a group in Germany. And they went, so we're moving from the back of the brain into the middle of the brain to look at hippocampal dependent regions of processing. And the task they use is kind of the old concentration game that we had as kids, where you had the cards and they were all laid with their backs to you, and then you could turn them over and see which card was where. You had to pick two cards that were the same and figure out what regions of space they're in. Well, so when you're doing this task, they also, while you were doing the task, they would give you a little rose scent. So while you were learning this particular task, you were also associating doing this task with the rose scent. Then people would fall asleep in the scanner, and and they were just basically scanning them again for that first little period of sleep. And during that period of sleep, they would either play a rose scent or some sort of non-odorous vehicle scent, something or other. And the idea is that could you remind the brain of what they were doing earlier at encoding, and then see if the same regions were active during encoding, were also active during this sort of reminder period, or this reactivation period, and how did that influence performance? So here's all the different experiments that they were doing, but we're just going to focus on just the fMRI study. So the basic finding, the behavioral finding is here, which is that if subjects slept after the training, and they slept and then they were given the odor, they actually showed greater amount of recall of those card locations only if they slept. So if you were given the odor while you were awake, it didn't actually influence you. You didn't show as much learning, but if you were given the odor while you were sleeping, somehow that triggered this idea of replay during sleep and increased your consolidation processes for just that task. And they had other sort of control tasks where they didn't show these performance changes. So now, in order to look for what regions of the brain were active both during encoding and during the consolidation period, they gave the odor during the encoding period and also during the consolidation period, which either was wake or sleep. And what they found are two regions of hippocampus that were more active during this period of sleep, as well as during the period of encoding. And that was the left anterior hippocampus and the left posterior hippocampus. So that was again the same idea of, which was previously shown in the visual cortex, which is that if you activate the process of consolidation with this odor, you're going to activate the same brain areas, and those areas are going to be pertaining to sort of memory-related brain areas. So then they got even fancier. And they decided to look at this idea of interference. Because when you reactivate a brain area, when you reactivate a memory, that memory becomes more labile. Suddenly you can change it from, we know from reconsolidation literature that you can actually change what a memory is once you sort of reactivate it, because then it becomes labile and you can switch around what people's memories actually are. So they decided to test this idea and see whether when things get reactivated, when a memory gets reactivated during sleep or during wake, does it become more labile and more susceptible to interference, or does it become resistant to interference, depending on whether it was done during wake or sleep. So the way they did that is they had a learning and coding task, and they gave the odor during wake or sleep. And that was followed immediately by interference, an interference test where they had totally different combinations of targets, locations. And then they looked to see what happened in retrieval for the original task after the interference task. And what they found is something quite interesting. First, they found that during wake, if you had reactivated the memory with the odor, you would show interference. So that's for the first task. So they actually showed that during wake you can lose, and that's right here, you can lose your memory for what you just previously learned because you interfered with it by reactivating with this odor. However, if you reactivated that memory during sleep, what we found is actually the opposite here, is that the odor memory from prior to sleep, the first encoding session, was strengthened and more resistant to interference than this vehicle, than the control condition. Meaning that sleep causes a stabilization of a prior memory and makes it more resistant to interference, rather than if you do the same interference during waking, the memory becomes actually more susceptible to interference. So then what happens when they brought this study into the scanner? They said, well, can we find the network of activity during both sleep and wake that is responsible for this kind of increasing of interference or increasing stabilization? Oh, it's noon. I'm sorry. Okay, I'm going to give you this really quick. Okay, what they found is that in the wake interference condition, the lateral prefrontal cortex seems to be most increasingly greatest association with this interference and decreasing memory. Whereas in sleep stabilization task, they found that the anterior hippocampus and the retrosplenial cortex and other areas of temporal cortex were associated with sleep stabilization. So the summary is just that rodent models show replay of wake neural sequences firing during sleep. We know this. In humans, fMRI activity during encoding is reactivated during sleep. We found this in perceptual learning for V1, declarative memory in the hippocampus. And I'm not going to show you this study because it's really, really interesting. I'm really sorry I don't have time for this. But yes, you can tie this reactivation to specific events during sleep using kind of an event-related analysis, looking at whenever there's a sleep spindle, let's concatenate all these sleep spindles and see what's happening during the encoding, during consolidation, during this particular sleep spindle. And they were able to find these really beautiful sleep spindle-specific learning brain region activations. And I just want to end with basically just, the wish list thing. So just to say, so the one thing is that none of these studies showed that the activity during reactivation periods, during sleep, was correlated with an improvement in learning. So even though we're showing that there's these interesting kind of things that look like this recapitulation of the memory encoding, none of that has been shown to actually correlate with improved performance at retrieval. So it's not really clear what that activity is, if it's not really related to performance changes. That's one thing. Another thing is, one question is, can we improve the temporal dynamics of fMRI methodology to better track the EEG components online in a sort of, because that would allow us to then start to do some really exciting things to see what's happening, moment-to-moment fluctuations of EEG and fMRI. Right, and then also to look at what is the network of brain activity associated with consolidation, because we know that there's all sorts of interesting network changes occurring across different sleep stages, and how can we then learn about consolidation periods by associating these neural network changes in sleep with what we know from memory. And actually on this last word by Busaki, which I think is also an interesting way of thinking about it, which, you know, as I feel like electrophysiology kind of leads the way, the question, what he usually says is, many methods can provide insights into the operations of the brain, but in the end, all these indirect observations should be reconverted into the format of neural spike trains to understand the brain's control of neural behavior. So the big question for me is, what is the association between all these bold signals that we're showing and this original data that shows that we have this neural spike signaling changes from sleep? So that's it. Thank you very much. Sorry.